Alrighty, thank you everybody. It's my distinct pleasure to introduce Dr. Mihaylova from Ohio State University. She is actually went to Ohio State as an undergrad, where she did some really interesting research there, getting a second author on a publication, and has had a tremendous career since then, working with Dr. Ruben Shaw for her PhD at the Salk Institute, and then working at the Whitehead Institute to try and understand how diet interacts with nutrient sensing in the gut and other tissues. And now she's back at Ohio State, where she promises that she focuses most of her anger at Michigan on that.
Can everyone hear me? Okay, hopefully it's the case on Zoom as well. Well, thank you, Dudley, for the really kind introduction. Really excited to be here. It's actually my first time at University of Wisconsin. And yes, no hard feelings from any Big Ten pseudo rivals. We do take most of our anger out on Michigan. But very excited to share with you today some of our previous findings from my postdoctoral training and kind of how we are thinking about ahead now with elucidating further the nutrient sensing and utilization in the mammalian intestine, particularly focusing in the crypt, which is what you're, sorry, I'm going to move this over because I don't think I'm, okay, I think that's better, where we're trying to elucidate better nutrient sensing and utilization in intestinal stem cells, which are located here in between these large Paneth cells. And early on, when I was starting my career in nutrient sensing and metabolism, and particularly in the focus in the gut, we were really interested in understanding how much of the microenvironment do the intestinal stem cells sense, and how much of it is communicated through other signals, hormonal signals, and paracrine signals from other cells. And I'll talk a little bit about that and some of the really nice work that came out of the labs of others that have parsed this out. But basically what our lab right now is currently focused on is trying to understand how nutrients are being sensed in the intestine and how different types of nutrients are being sensed. So how do epithelial cells respond to nutrient deprivation and also overabundance of various different types of nutrients? In particular, also we're interested in understanding how chronological aging impacts nutrient sensing and whether some of these nutrient sensing pathways become more sensitized or desensitized with aging. And I'll talk a little bit about that in a couple of slides from our previous findings. And then more recently, we're becoming very interested and kind of cannot really ignore another component of the GI, this microbiome component, which clearly has its own very complicated sensing and production of various different metabolites, which are also utilized by the epithelial and immune cells. And so the gut is a very interesting sensory organ and orchestrates a lot of different functions. And so something else that we were very interested early on was trying to understand the influence of diet on longevity and on aging. And this was, again, influenced by many of the wonderful labs that I was surrounded by during my PhD in the Salk Institute, as well as also during my postdoctoral training. As we all are aware, across many different model organisms, caloric restriction really prolongs the median and maximal lifespan across species. So if you deprive C. elegans from bacterial culture, it would lower that down, or lower also, sorry, this is kind of not working. Okay, lower down yeast extracts or chow across some of the different backgrounds of mice, you see these extension of lifespan. And so we were very interested, along with other groups, what are really kind of the more tissue-specific effects of nutrient deprivation and how nutrients are really sensed in a tissue-specific manner, and which tissues are important for propagating these effects on health span and lifespan. And so, of course, I have to also dedicate a lot of this inspiration to Drosophila work and work in other model organisms, because they very nicely showed this connection between diet and its influence on aging, and also on homeostasis on various different tissues within these model organisms. So particularly here, I'm showing you the effects of aging on the epithelium in Drosophila, which was very well characterized by several groups, showing that with age, you actually get hyperproliferation, which is a little bit different than what we see in the mice, as I will describe in a couple of slides. But also as you age, you kind of lose this epithelial integrity in the Drosophila, and this leads to this really interesting findings of when you actually dose the flies with a dye that can go through the epithelial lining, you get these smurf flies. So this just really shows you that the integrity of the gut is compromised as the flies age. And really important studies also linked and connected the ability of this to be prevented by, again, dietary interventions and mitochondrial function, improving the mitochondrial function. So we were very interested to understand how these effects were being also, whether these effects were also seen in the mammalian tissue. And of course, today, most of my talk will be focused on mammalian physiology and aging and metabolism in mammalian tissues. And so not to belabor this, but we are very well aware that we have a very large increase in the aging population. And with that come a lot of age-related diseases and morbidities. And so, as we have increase in aging population, there's increase in cancer, Alzheimer's, a lot of metabolism-driven diseases. And so we're very interested in understanding how we can slow down and perturb some of these. And again, as I mentioned, a lot of work and seminal work has been done on how diet really protects and slows down the occurrence of a lot of these types of age-associated diseases. And so, of course, aging is like a very complicated accumulation of a lot of different deregulated processes. So this was a review that came almost 10 years ago, was modeled after the seminal Hallmarks of Cancer review from Doug Hanahan and Bob Weinberg. But this Hallmarks of Aging review really nicely highlighted just the complexity of the all different types of processes that become deregulated with age. And so you have some that are seemingly quite irreversible. You have genomic instability leading to a lot of mutations and alterations in the genome integrity. You have telomere attrition, epigenetic alterations, and a lot of more transcriptional noise with aging, loss of proteostasis. And then also some that are a little bit maybe more seemingly malleable, such as deregulated nutrient sensing. One can hypothesize that perhaps you can target these with different diets and see whether you can improve the ability of these nutrient sensing pathways to regulate and function better in the aged tissues. And that's one of the interests that we have. But again, contingent on some of the other issues that we also face, whether this is contingent on mitochondrial dysfunction, that's again something that we're looking into in the lab. Some of the metabolic consequences that we're seeing of aging potentially could be also irreversible because of this particular aspect. And then downstream of that, there are, other consequences that can lead to like more catastrophic events, such as cellular senescence, exhaustion of stem cells, and then altered cell-cell communications. And so again, all this to say is that it's quite complicated to tease out. And also, we are interested in another aspect on the other end of the spectrum. When you look at cancer, there are a lot of actually parallels between aging and cancer. There are a lot of the regulation of homeostatic pathways and processes. And so not surprisingly, the regulated cellular energetics and metabolism is a hallmark also of cancer. And this was, again, very nicely shown early. Now it's more established hallmark, but something that's been known for quite a long time from even Warburg's experiments in the 1920s. And so when I joined my postdoctoral lab, I came from a signaling lab. So some of the first things that I was doing was really looking at nutrient sensing across several different tissues that I wanted to work in. When I did my PhD, I actually rotated in a stem cell lab and also in the metabolism lab. And I ended up joining a metabolism lab, which I was very, very excited and happy about. But I was also very interested in sort of the metabolism and the physiology of adult stem cells, and connecting this to aging and seeing how they really change over time and how sensitive they are to the microenvironment and nutrients was something that was an interest of mine for some time. And so I had a little bit of time to explore during early days of my postdoctoral training, and looked into various different tissues to see, first of all, how responsive they are to nutrients in respect from young and aged animals, but then also more on the different types of cells and how they're responding to nutrients. So if you just take, obviously a piece of tissue and you look at the signaling, you can see most tissues respond quite robustly to the deprivation of nutrients. And then in growth permissive or these refed conditions, they turn on all these pro-growth signaling pathways. And I did this, probably was like in the first year that I was in the lab. I took out these units, these crypts from the intestine, dissociated them, and did a Western blot, and looked at how effectively they were responding to nutrient deprivation, but again, in a very holistic way of just the entire crypt. But then, and again, if you look at more spatially, you can see now and appreciate that there's heterogeneity in terms of nutrient sensing and responding. I don't know how well this is showing up, but even in the liver, which is something, the organ that I really, really loved during my PhD and worked quite a bit. And, I will say a lot of people were like, oh, the liver is kind of boring. It like has one cell type and they all are responding the same way. No, that's actually quite not true. They do have this gradients of nutrient sensing, and they're gluconeogenic and glycogenolytic hepatocytes. And so where you're getting your tissue from and what region actually also matters. When it comes to the intestine, what was really interesting is that you saw like globally the response, but there were some cells that were responding better than others. And actually some of that was a work that was done at the time in the lab by two postdoctoral fellows, Pekka Kadajisto and Omar Yilmaz, who were looking at the response of Paneth cells, which are the support cells of the stem cells, and how they're responding to nutrient deprivation and caloric restriction, and how those paracrine signals were propagated and influencing stem cell self-renewal. And I'll touch a little bit briefly on that in the next couple of slides. And then another tissue that I was looking into at the time, which I thought was really cool, was the skin and the hair follicle. Again, it's enriched in adult stem cells. And I was interested to see how these stem cells really sense their microenvironment and how they make decisions about elongation and kind of how that affects the hair cycle overall. And since then, very nice studies have shown that actually dietary deprivation can really affect hair follicle cycle and sort of the amount of hair follicles and density that you have in a mouse, which kind of makes sense because it might be also affecting thermogenesis and sort of heat response as well. So today I'll mostly focus on the intestine, but just kind of wanted to give you a little bit of background of my background and what really interested me in these questions. And so, as I mentioned, when I joined my postdoctoral lab, two postdocs were working on this really interesting question of how caloric restriction was being sensed in the intestine, and their focus was primarily on these Paneth cells, which are located next door to the stem cells. The stem cells are very small. They largely are hard to actually even image very well in the intestine, but these huge Paneth cells next door to them are very important. They secrete a lot of paracrine signals and are ensuring the self-renewal and differentiation, the right amounts of that in the intestine. So they're very important for the niche. And what they discovered was that in this adaptation to caloric restriction, the Paneth cells were secreting, first of all, they were downregulating mTORC1 signaling, and then they were upregulating cyclic ADPR secretion to then enhance stem cell self-renewal and dial down differentiation in the state, which was quite interesting. And the question that I was interested in at the time to kind of complement some of this work was to really look and see whether the intestinal stem cells themselves are sensing independently this nutrient changes, and how do they metabolically rewire to this nutrient deprivation. And so the approach that they were looking was the caloric restriction, which is kind of like a long-term adaptation. What I was interested to see is more in acute day-to-day changes. So the regimen that I chose was a 24-hour fast. And also as an aside, I wanted to mention, and this is something probably you have heard from Heidi's work in Dudley Laming's lab and others, but it's important to know that caloric restriction historically was done in a means where the mice were fed just once a day, and then they essentially were fasted for 22 hours or more. And so this really fasting phenotype was important to further explore. And I'm so happy that so many people are looking into that now as a component of caloric restriction to shed more light of that particular contribution of the CR phenotypes. So at the time, I also wanted to give you a brief background. Probably most of you are familiar with the intestinal hierarchy and the architecture, but the mammalian intestine and epithelium is highly, highly regenerative. These terminally differentiated cells live only for like 3 days or so. And so all of this renewal is really dependent on the stem cells that are at the bottom of the crypts. They're marked by these LGR5 receptors that are really highly expressed. They're basically Wnt amplifiers, and there's a Wnt gradient. So there's a higher Wnt secretion and accumulation here in the crypt, which really keeps the stem cells in a stem cell-like state. And so self-renewal of the stem cells obviously is essential, but they also give rise to all the progenitor cells. So now with the advent of all the single cell technologies, we have more and more flavors of these early progenitors, more on the secretory or absorptive path, and we know more about what some of these key transcriptional regulators are there. But basically these progenitors then differentiate and give rise to secretory or absorptive cells. In the secretory compartment, we have mucus producing cells, so MUC2 cells producing goblet cells that basically coat the entire epithelium, and that prevents from the bugs being close to the intestine, also lubricating that and preventing abrasion. You also have enteroendocrine cells, which are really important to secrete, basically, sorry, to secrete hormones and to really kind of orchestrate the sensing of different nutrients in the intestine. And also you have these really interesting tough cells that also are chemosensing in nature. And so altogether, this works to regenerate the epithelium every 3 to 4 days. And of course, these epithelial cells are not, living there by themselves. They also are in close proximity to fibroblasts and other types of cells, such as immune cells, which sample the microenvironment and detect the type of metabolites and also bugs that are present and form other decisions in the intestine in case of infection and so forth. And so very complex, interesting system in general that really, really is interesting to study. And so, as I mentioned, some of the questions that I had early on was trying to understand what drives these age-dependent changes in the stem cells. So there were some really nice reports, morphological reports early on suggesting that there is depletion, and also the intestinal stem cells are less in number, and there are some morphological changes that are happening in the intestine. There's also less regeneration in response to insults like radiation. And so we wanted to look a little bit deeper into that. We wanted to also see how nutrient availability and utilization is affecting intestinal stem cell function in the gut, and also repair to some of these insults. And also, our hypothesis was that some of these insufficiencies that are coming during aging are dependent on metabolic perturbations. And so we wanted to look more unbiasedly what some of these are using various different approaches, which I'll talk about a little bit today. And a lot of our work is done using these really nice reporter mice that were generated by Hans Cleaver's group. So basically you have in these very high LGR5 expressing cells, a CRE-RT2, as well as a GFP reporter. So what this really enables you to do is to take these cells and really sort them and parse them out on the single cell level. So you can first mechanically and chemically dissociate the entire crypt epithelium, and you can take these units, you can embed them in extracellular matrix, also known as matrigel, and actually there's really nice mechanosensing that happens there. And the crypts will fuse, and then over time, they'll regenerate these crypt domains. So you kind of you have basically these enteroids or organoids that very closely resemble the architecture of the intestine itself. And as I mentioned, you can take the GFP high expressing cells or low expressing cells, which are more on the progenitor side. You can further dissociate this entire crypt, and through FACS sorting, really bin them and study their, basically ask questions on how they're behaving on a single cell level, and whether some of these changes are coming based on what they're sensing versus what their neighbors are sensing, and ask questions about that through these mixing experiments. So actually you can take intestinal stem cells from one particular animal that's either ad libitum or fasted or young or old and mix them with Paneth support cells from a different animal that is just like ad libitum and ask questions, are these signals really coming and propagated to the intestinal stem cell, or are they being propagated through like a neighborhood cell? And this takes a little bit longer to form. So these organoids, you know, obviously they start from like a doublet of a cell. They actually find each other in the matrigel. It's really, really neat. And then they form the organoids here, as you can see, over the course of a week or so. And as I mentioned, these very nicely resemble what you see in the gut. So you have the Crypt domains, you have the same architecture of the alternating stem cell Paneth cells, and you have the enterocytes, as well as enteroendocrine cells, goblet cells. And you can actually modulate the composition of these cells based on what you place in your media as well. You can have a composition that's more stem cell or more differentiated based on what you put in the media. So that's a very important point to what exactly is in your media. So first question that we wanted to address was what drives these age-dependent changes. And so we enumerated from aged animals. Again, these animals were aged by the two postdocs that were in the lab previously. And in collaboration with them, these mice basically were between 18 and 24 months, and we were able to collect these cells and see basically are there the same number of stem cells based on the GFP marker. And what we found time and time again was that there was indeed a decrease in the number of aged GFP positive stem cells. What we also wanted to test was their functionality. So we again took the stem cells and mixed them with young or old Paneth cells and asked questions about how functional they are. And as you can see, the most functional kind of pairing is not surprisingly between the young stem cells and young Paneth cells. And then as you add in old stem cells, you actually have a decrease in this clonogenicity, again, saying that there's some defect happening in the intestinal stem cells with aging. What was really interesting also was the different composition changing from proximal to distal, kind of regionality in the intestine on the secretory cells. So again, we and others have also reported this, see an increase in Paneth cells in the distal area of the intestine and maybe some changes in goblet cells. Others have also reported there's increase, and people think that that might be due to perturbed microbial composition with aging or response and kind of containing that microbial composition with age. And so that's a very interesting aspect as well. We also wanted to see how, again, in vivo proliferative and functional these stem cells are. So we did some lineage tracing experiments where we induced a marker with tamoxifen driven by LGR5 to be labeled in the stem cells. And then 3 days later, about the time that it would take for these progenitor cells to get up on the epithelial escalator, sacked the mice and also looked at how much of these villi were labeled for TD tomato. As you can see, this labeling is not as robust in the aged animals, suggesting again that these are less proliferative. And we also did some challenges where we induced damage using radiation, and we saw that the aged animals are not as efficient in regenerating their gut epithelium as the young ones. So collectively, this told us that there is something kind of wrong with intestinal stem cells as they age. And so what really is going on there? So as I mentioned, we can take these Crypt domains and we can embed them in matrigel and enumerate how many of them can actually form organoids. This is a test of how functional the stem cells are. And again, we were able to see that per unit Crypt plated on the same exact amount between young and old, the aged ones always gave us a lot of, let me see if I can use a cursor, a lot of Crypts that didn't really take off. And also if they did, they weren't very well differentiated. They didn't form these new stem cell rich domains. And so we hypothesized that perhaps there was some metabolic defect, right, that might be happening in these cells. So we tried to make a number of different approaches to really test this. Now, I will tell you that I'm very, very interested in finding out what is the metabolic characteristic of the stem cells, but it's something very hard to answer because they're, single cell metabolomics is not quite there yet. And the metabolites are very labile, so you have to work really, really fast in order to address that. And so, we tried some creative approaches in trying to understand, first of all, in the crypt as a whole, how the metabolism of these cells is changing with age and also in response to different dietary states. And so one of the approaches we came up with was to look at labeled either glucose or fatty acids and see their uptake and their ability to uptake, whether it's equivalent between young and old, and also their utilization by tracing these carbons through the beta oxidation and also for glucose through the glycolysis, and then looking at amount of lactate and so forth. And so what we found, and this we haven't really fully published, again, we really, really want to verify it in multiple different ways, is that the aged crypts appear to be less metabolically active in general. And so we have, less utilization of palmitate and less utilization of glucose. I thought, well, maybe they're more glycolytic. Maybe there's some compensatory, mechanism. And we've seen that before. If we knock out like a transporter for fatty acid, transport basically CPT1A into the mitochondria, we actually sometimes see concomitant increase. The cells will adapt and they will bring in more glucose to utilize because they have to proliferate in the young animals. But in this particular case, just looking at wild type aged animals, we saw that there's less utilization of glucose and fatty acids. And of course, we haven't ruled out some kind of supplementation from amino acids and so forth. This is not by any means all exhaustive, but this was an interesting aside that I wanted to share. And this kind of nicely fit with the phenotype. We were seeing that there's less turnover in the intestine, and also there's less proliferation. So here, these animals were injected with BRDU. And as you can see, we see less labeling in the aged animals compared to the young in the stem cell compartment here in the bottom of the crypt, and as well as in the transient amplifying cells that are found here on the top of the crypt. And so very nicely, about maybe now 2 years ago, a paper came out from Bert Vogelstein's lab that they suggested that indeed they see also less cell division rates and that they decrease with age. And the hypothesis they put forward, which I thought was really interesting, was that there is perhaps an age-dependent deceleration in cancer incidence. So this proliferation is sort of like a protective mechanism potentially for the gut. Whereas if you remember in Drosophila, they're actually with age see a hyperproliferation phenotype, which is quite interesting and a little bit different between what we see with mammals. And so just a little bit of summary for this part so far. So some of the age-dependent changes that we see in the intestine are described here that I went over. And then there are other types of changes that obviously can occur as well. And we're very interested in exploring them a little bit further unbiasedly at the moment. Hopefully we'll have some updates on that in the upcoming years. But many papers have also very nicely shown other defects. Some are more focused on the metabolism side. So there's, again, defects in NAD metabolism that others have shown. More unbiasedly, Alessandro Ori's group have reported there's changes in, again, metabolic enzymes between young and aged animals. And then a lot of people have reported the other very important factor for stem cells, that Wnt signaling is also decreased. So there's, sort of a synergy between perhaps decreased Wnt signaling as well as, some metabolic perturbation, and maybe there's even crosstalk between the two. So there are a lot of probably additive effects that are happening there. There's not just probably one. And so the other question that we had, as I mentioned, is how nutrient availability and utilization is affecting intestinal stem cell function in the context also of gut repair. So as you recall, early on, I was showing you this Western of just the entire crypt ground up and looking at Western blots of pro-growth signaling. So in the fasted state, the crypt is kind of shutting off all of these pro-growth pathways. And then just 2 hours later of refeeding, it's turning back on all these pro-growth signaling pathways. And this can also be nicely seen here more on the spatial side with IHC looking at phospho-S6, which is downstream of mTORC1 signaling. And you can see again that in the fasted state, not all the cells, but most of the cells shut off this pathway and then turn it back on. Curiously, there are a few cell types that are potentially still keeping it on, and it's very curious why that is and what the mechanism there is. We're suspecting that might be enteroendocrine cells, but For the majority, the Crypt is very responsive to that. And so I remember I told you I was interested in this short-term fasting. So one of the first experiments I did was this fasting, refeeding, and experiment. So I fasted the mice, and then I took out the Crypts and I embedded them at equal amounts in the matrix gel. And you always kind of remember those striking experiments. I looked at it the next day, and there were like twice as many organoids in the fasted animals as in the libidum. And I was like, wow, I probably messed up like the samples or something. But I repeated this a few more times, and every single time I was getting the same phenotype. And now others have also shown this nicely, which I'm very happy that it was independently verified. But somehow this fasting in the animal really primed these cells to be more clonogenic and to really give rise to more organoids in the culture. And I think the way we presented this may have been a little bit confusing to people, because if you think about fasting, you'll say, well, just in the previous slide, you showed us that all the pro-growth pathways are like downregulated. So what is really happening here? Why are they more proliferative? But you have to remember that this is like actually nutrient-rich media. And so these cells kind of become primed. They really do become primed to, I think, uptake more nutrients and really kind of go wild in this media, if you will. They really proliferate much faster and generate more organoids per Crypt unit. And this is quantified here. And I'll kind of build up a little bit on what we've followed up since then, what we think might be happening there. We also tested this on the, again, cell-specific question. So is it really coming from the stem cells or propagating by their neighboring cells? And in order to do that, we again sorted out equivalent intestinal stem cells as well as Paneth cells that were just from ad libitum mice, and then mixed them and asked questions. But what was really curious is the fasted stem cells themselves, without even the Paneth cells, had this increased clonogenicity here. And when you added just ad libitum Paneth cells, because they do secrete quite a lot of growth, pro-growth factors and also Wnt, they do increase their clonogenicity even further, again, the fasted compared to the ad libitum here, suggesting that there is something that is changing in the stem cells that's priming them to be better and grow better. So what could that be? We kind of took an unbiased approach by RNA-seq. This was before kind of the single cell days. So we sorted like 50 to 100,000 cells from the GFP positive stem cells, progenitor cells independently, and then asked what are some of the most upregulated and changed pathways there. And what really was striking was this phenotype of increase in PPAR targets and fatty acid oxidation. So these cells really sensed the changes in their microenvironment, and they switched their metabolism from more glucose, perhaps utilizing, to more fatty acid utilizing. So in a way, this is not quite so surprising. There are a lot of tissues that do that. So when you have nutrient repletion or carbohydrate limitation during fasting or during exercise, for example, a lot of our tissues go to an alternative source. First, the use of the glycogen, and then they start through lipolysis getting fatty acids from our storage tissues in order to activate this fatty acid oxidation and to just get through with generating more energy there for survival. And also to be, transparent, if you were to sac the mouse and take the in vivo tissue, you will actually see the suppression of cell cycle genes. So, it's something about these cells getting primed, and when you are culturing them in the matrigel, in the full growth media, that allows them to regrow. So in a way, this is like a refeeding response. And if you really think about it, that makes a lot of sense evolutionarily as well. You probably don't want to use up your energy to make more tissue and for more tissue that is absorbing no nutrients, right? So you're probably kind of sitting there a little bit primed, ready to go when there is more nutrients hit again, when the conditions are permissive for growth and for proliferation. So we started focusing more on this like refeeding response now. So what we think as a summary is that in the fasted state, these fatty acids are released, so they're taken up for, again, purposes of energy generation through beta oxidation in the intestinal stem cells. The major players here are these PPAR genes, which are nuclear receptors. They actually bind and sense these fatty acids, turn on genes that encode enzymes that will uptake then the fatty acids and increase beta oxidation and utilization of these fatty acids. And then here's just an example of one of them, CPT1A, which is a critical player into bringing in the fatty acids into the mitochondria. We also used our metabolomic approach in the Crypts to really kind of show proof of principle for this. So we had from Peter Carmelier, the CPT1A flox mice that we cross into the Villain-Cree, where we can inducibly generate a CPT1A knockout in the intestine and show that, first of all, it was a very nice deletion using Villain-Cree here. These are whole Crypts again. And then we took out these Crypts and we traced 13 carbon label palmitate to see what the utilization of this was in these cells. And we were able to see that there was a significant suppression of the byproduct that is generated and exported as a result of beta oxidation in the CPT1A knockouts. Again, proof of principle, and also M2 labeled citrate. Also we were able to show that blunting CPT1A and the import of fatty acids into the mitochondria basically decreased this fasted induced increase that we reported on following like a 24-hour fast. So again, this particular aspect downstream of the fasting, taking up the lipids and utilizing them for beta oxidation was integral for the fasting responses that we observed in the culture. And over time, we were able to see that there was a decrease in stem cells. So if you kind of prolong the deletion in CPT1A, we saw less LGR5 positive stem cells in the bottom of the crypts. So what we think happens here again is that in the fasted state, the PPARs are activated. They take up more fatty acids from the microenvironment and then utilize them for beta oxidation. And now we're also looking into the upstream regulation and seeing which PPAR is really responsible for this. So we have some single and double knockout PPARs, PPAR alpha, PPAR delta. Those are the ones that are expressed in the intestine, and seeing how those affect the PPAR target. So again, in the PPAR alpha alone, we see that with fasting, it's CPT1A is induced, and in the knockout, this induction is suppressed. And so similarly, HMGCS2, which is another important component of ketogenesis in the intestine. And this again is seen here by this Western. We also saw a decrease in chronogenicity in the PPAR knockouts. Again, in the fasted state, we don't see such a robust response, but again, there probably is also other components to this piece of the puzzle. What was really interesting though, was showing that enforced fatty acid oxidation actually improved proliferation. And so again, as we showed earlier, there's a decrease in this organoid formation in the age, this defect. But now if you treat animals with GW, which is an agonist for PPAR delta, you actually can force them into uptaking and utilizing more fatty acids. You kind of switch pharmacologically the cells to become more fatty acid oxidizing, and this increases their proliferation and also improves some of the aging phenotype that we observe. It's probably not the full picture. And it also improves their proliferation. So again, I think this was a very interesting aspect because this is not a fasted state, right? So you're kind of forcing the cells to use more lipids, but they also have access to glucose as well. So I think this is sort of the prime ability for them to really have like high energy state and also use the glucose here for proliferation, for making more lipids and membranes for potentially, and not really be shunting the lipids more towards beta oxidation. And so this is sort of the follow-up of what we're doing currently, trying to really tease out that aspect of lipid utilization in these states. And with that always comes the question, well, is it really good to force proliferation on an aged tissue that potentially could be, full of, some mutations? And so that answer is to be determined. But just in the regenerative aspect, we think that this is improving the regeneration of these intestines in the aged mice. So as I mentioned, we haven't formally really fully shown this in the intestinal stem cells because again, we can't really do yet like robust single cell metabolomics on the intestinal stem cells. But what we are seeing is there's a decrease potentially of FAO. So this comes to the question of, is it really driven by transcription? We don't really see like transcriptional changes as much, but maybe it's more like on the function of the mitochondria itself. So we're doing a lot of functional tests to test whether the mitochondria itself is less functional. Maybe that's what's driving some of these phenotypes as well. And we can, as I mentioned, restore the proliferation using GW, but this is more on the kind of functional side of things, where we can increase, again, the uptake and utilization of fatty acids by driving up this fatty acid oxidation phenotype pharmacologically. And this is just showing the metabolic aspect of that, that indeed it is more active when you're treating the animals with the GW. Now, another thing to really think about is actually the availability of these lipids to the cells and whether those change with age too. Something that we did notice was that in fasted animals, there was a decrease in availability of palmitate in the serum. So it could be that there's also some additive effect of that, that maybe they're not really having as many available fatty acids for them to uptake, and maybe that's part of the problem as well. And when you supplement it in culture, it actually improves, again, the aging phenotype to a degree. So that's another thing to think about. Where are these lipids coming? What are the demands of these cells? Because they're very, very highly proliferative cells in our bodies. So a little bit different than like terminally differentiated cells. So this was our model in terms of this study. And so we looked at, basically we think that basal in young animals, they're probably utilizing basally fatty acid oxidation and glucose, and that's sort of their really best state. But with aging, this is becoming perturbed, still to be determined why. Is it just all environmental, or maybe it's intrinsic and extrinsic? And then fasting really primes these cells to uptake more of these environmental lipids, but also when you refeed the animals, really take up also the glucose and everything else that's available to them, and this kind of spurs this pro-growth phase. And again, as a whole for the intestine, that makes sense, because when you have nutrient availability, you want to proliferate, you want to elongate your villi, you want to increase the ability to probably absorb all these nutrients in the state now that that is replenished. And we can mimic some of these aspects pharmacologically, but we wanted to look a little bit deeper, as I mentioned, and this is something that we've been doing since I started my own lab, and trying to understand what other aspects of this lipid metabolism are relevant. So we went back to our RNA-seq data, and we looked at what other pathways are being implicated in the fasting refeeding phenotype, particularly that feed into the fatty acid oxidation and interact with fatty acid oxidation. And what was really curious were these steryl desaturases that are in steryl-CoA desaturases that are regulated very robustly with fasting and refeeding. And probably all of you are very familiar with what they do. Dr. Ntambi is here at the university, and he's done a tremendous work characterizing them over the years and really showing how important and integral they are for lipid metabolism in various different tissues. And so we wanted to explore a little bit better what their role is. We looked at basically where they're expressed and how much they're regulated, again, confirming some of the RNA sequencing data. So interestingly, they were both, so there's 4 isoforms, but in the gut we see only 2. So STD1 is the most prevalent one, and STD2 is another one that we see that is sort of more found in the crypt, which is quite curious. And as I mentioned, we see them responding quite well with fasting and refeeding. So as you can imagine, in a state where you're not permissive to grow and proliferate, you probably want to shut down this pathway that is now, involved in de novo lipid biogenesis and making more membranes and so forth. You want to actually shunt your lipids more into fatty acid oxidation, more mitochondrial metabolism. And that's what we think is happening here. And then this is just some very, very recent data. So in fact, some of it was sent to me in the last couple of weeks, but from my graduate students. And so what we were trying to show here is that in the fasted state, again, you have downregulation of AKT signaling, pro-growth signaling, and you actually have a decrease in STD levels. You have an upregulation of other fatty acid oxidizing genes, but you have the downregulation of de novo lipid biogenesis genes. And as I mentioned, these enzymes are very integral in converting saturated fatty acids into monounsaturated fatty acids. They're important for mitochondrial function, ER integrity, membranes on the plasma membrane, And then they're also regulating downstream of that lipid droplet formation, protein acylation, and all other very important aspects of sort of lipid homeostasis and other processes in the cell. So two of the hypotheses that we had was that potentially this refeeding insulin high state, or maybe even overabundance of lipids and glucose, can really upregulate these desaturase activity in the intestinal stem and progenitor cells, which will increase de novo lipid biogenesis in this pro-proliferation state. And then also these Western diets can mediate STD upregulation, further influence proliferation. So a lot of papers, including our collaborators from before, have shown, and many others, that these Western diets really increase proliferation in the crypt. And so what is really driving that? We think that this is probably part of the puzzle. There's an increase in STD activity and probably increase in lipid membrane formation, and which is necessity for proliferating cells. We also wanted to see whether inhibiting the STDs in our sort of fasting refeeding phenotype in culture really had an effect and blunted some of these effects that we previously reported. And what we were able to see is a dose-dependent decrease in, again, clonogenicity using an STD inhibitor, which is supposed to inhibit both STD1 and 2. So that was quite interesting. And now we've done some variations of different diets, trying to really tease apart and see what substrates are driving these phenotypes. So in a diet that has, these are sucrose matched diets, so that's kind of an important component, and also protein matched. But when you increase saturated fatty acids and have a decent amount still of carbohydrates, we really see an upregulation of these STDs, and we also see an upregulation of proliferation. So this is sort of, again, kind of our refeeding state here. And what's really curious, we haven't really teased this out yet, but we see that there's a downregulation of de novo lipogenesis. And so very kindly, we were able to obtain mice from Dr. Nantabi here and delete them specifically in the intestine. So when we delete STD1, we actually have a decrease of the de novo lipid biogenesis. And actually, I'm not showing this here, but we have an increase in fatty acid oxidation genes. So you have sort of like the energy there, but you don't really have the pro-proliferation phenotype. We think you need to have both in order to proliferate. We also have, with collaboration with Martha Belluri in Ohio State, and also more recently designed some of our own diets where we have fed mice saturated versus unsaturated fatty acid rich diets. And we've seen that in the saturated fatty acid diet, there's an increase in STD1 levels again. These are probably needed there to desaturate the saturated fatty acids so the cells can avoid lipotoxicity, but also to, again, give rise to like all the downstream lipid components that are necessary to support proliferation. But in the unsaturated diet, we don't see the upregulation. They probably, from the microenvironment, take up these unsaturated fatty acids, and that's probably sufficient. So again, we're doing a lot of pharmacological inhibition and genetics to really tease these apart now. This again is very, very recent data. And as I mentioned, increasing these lipids will drive proliferation. Others have reported that previously as well. And more so studied is in a case of a cancer. So there's quite a few different types of cancers that have been shown by many, many different groups. A lot of them, albeit, have been shown more like on a ex vivo setting, but these STDs become very important for proliferation and very hyperactivated in a lot of cancer cell types. And some of it might be driven even by the type of mutations they have. So if you have like pro-insulin signaling, this will be regulated downstream of SRBPs and will be probably turned on further on that basis. But what was interesting was when we tested STD inhibitors on APC null colon organoids, they are capable of, again, inactivating this like pro-growth signaling and pathway and kind of suppress the pro-growth of these organoids that are more in the cancer-like state and more pro-proliferative. And there have been some really nice and interesting studies that have also been done in the gut. Again, this is a study from Dr. Nantabi and collaborators, where they've shown a very interesting role here. So in this particular case, you actually have, I believe, a whole intestinal deletion of STD, and that is actually blocking the uptake and absorption of lipids and the desaturation of certain lipids in the villi. So this creates kind of a pro-inflammatory state, and which was really interesting is actually the lack of STD in the enterocytes here is really kind of driving the tumor phenotype. So you have to be really careful when and exactly how you're looking at this, because in this particular model actually was exacerbating the adenoma phenotype. We predicted in the stem cells that are starting the adenomas, if you perturb STDs there, particularly, you might actually suppress the growth. And that's our hypothesis on the generation of these adenomas, and we're testing that now. So just to, again, give another summary, some of their ongoing experiments and future directions are to determine further how dietary composition affects intestinal stem cells and their microenvironment. There's a lot of crosstalk with intestinal stem cells and other cells. And so we're doing some single cell analysis there, at least on the transcriptional side of things, to see what potential crosstalk is happening there. And there's some really nice computational ways of doing that right now, including cell chat and others, which I unfortunately don't have time to discuss today, but we're actively working on that. Also trying to determine substrate utilization in intestinal stem cells and their niche during early cancer initiation and progression, where you have these hyperproliferating phenotypes. What other intrinsic and extrinsic factors may contribute to intestinal stem cell aging? Again, I highlighted that there are quite a few different things to consider there. It's not probably just one particular aspect of a homeostatic mechanism that's off. So we're trying to stay as agnostic as we can, and also examining how diet composition and nutrients can influence the microbiome. So that's something more recent that the lab is looking into. Again, it's hard to ignore the bugs, which are really, really interesting and produce a lot of very interesting metabolites. And there's for sure a lot of crosstalk between what is produced by the microbiome and also the epithelial cells and immune cells there. And we also have some tool building that's going on in the lab. Just because we can't measure single cell kind of metabolomics, it doesn't mean we're not trying. So we're trying to do more in the 3D organoids. It's very challenging because, again, it's hard to normalize some of the effects that we're seeing. These things are grown in a matrigel, which is very protein rich. So with our collaborators in the University of Kentucky, Andrew Lane, Richard Higashi, and Teresa Fan, we're trying to come up with better ways of measuring the flux and the utilization of metabolites in the organoids. And this is just one of the trial runs that we were doing with glucose. So we've kind of worked that out, and we're doing other tracers at the moment. And then with this really wonderful collaboration that we've started at Ohio State with Amanda Haman's group, and particularly her postdoctoral fellow, Emily Scerera, and my graduate student, Kubra Kaya, and postdoctoral fellow, James Sleziona. They're very interested in looking at MALDI imaging and looking at lipid abundance that way. At least the lipids are somewhat stable. And so we're starting there, but eventually hoping to maybe look at smaller molecules, especially if we can label them and, you know, have a meaningful way of quantifying them and seeing how they're changing in response to these different effects. And you can get really, really neat images like this. And we will use pharmacology and genetics to try to understand what all these different lipid species are and how they're changing in response to our inputs. So just to summarize, again, we're building quite a few tools to really try to understand the consequences of this aberrant nutrient sensing, either with diet or aging or in cancer, and either also in homeostatic conditions, because I think it's really important to understand the homeostatic conditions before we make conclusions about age-dependent changes and cancer-dependent changes. We're now really interested in kind of spatial technologies. So we're trying to work out on some of those and integrate them with our other approaches in the lab. And there are a lot of people to really thank, including the brave graduate students, the first ones that joined the lab and the first lab members, as well as many very nice collaborators that we've been able to recruit and, very excited to continue our collaborations on the spatial aspects. Amanda Haman, as well as many collaborators at Ohio State that have helped us and given us their invaluable input. Dudley Lamming, always for a lot of advice and early day kind of bouncing off ideas, and always everything aging related, talking to him about is really helpful. Dr. Nantambi for all the mice and discussions that I hope we will continue to have on the STDs. I'm just excited about that line of work, and our collaborators at the University of Kentucky who are helping us on the tracers, and our former collaborators and mentors, and graduate students and undergrads that helped along the way. Also in particular, Chai Wei Chang, who really contributed a lot to the fasting story. And Peter Carmelier, Becca Catugisto, Maria Mas, everybody who's contributed to this work, and our funding sources. So happy to take any questions if there's time left. It's a fun aside.
So I might have missed, but CD1 seems to be correlated with CPG expression, the coordinated. So both of them are related by complex signaling?
Yeah, so that's a really great question. So one thing I didn't show was actually the effects of blocking pharmacologically STDs. What happens there is that the fatty acids are kind of shunted. I think there's like a feed forward with PPAR signaling. So they actually increase transiently on the transcriptional level, the CPT1A enzyme and others. But I think it's almost like a futile cycle in a way. I think it helps with like energetics and energy balance in the cell, but you still need, I think, the products and the fatty acids to obviously for proliferation to make the lipids that are necessary downstream for that. So if you block the utilization and their desaturation and so forth, and there's not enough from the microenvironment, you can, the microenvironment ones are shunted in, you block the intracellular utilization, you actually can increase the intracellular pool transiently, and then enough to drive PPAR dependent transcriptional upregulation of the FAO genes, and then increase those levels. But then how long that prolongs and like stays on is something to be determined. Obviously, we can't fast the mice too long. And after a while, you might kind of cycle out and have to adapt in a different way. You might kind of run out of the lipids. But it's a good question. And yeah, there's this kind of like transient bump of like a feed forward where you block the lipids and they upregulate the FAO. Yeah.
So clinical talk, I was curious, when you were discussing some of the stuff about the organoids and the crypts, I was just wondering, is there a difference between the organoids proliferation and from the crypts that could come from the different intestinal regions, such as proximal crypts versus the distal crypts?
Were there differences in terms of their clonogenicity? That's a really good question. I think other groups are looking a little bit more into that than we are, and we're starting a little bit. We haven't seen a ton of differences, but I think there might be some age-related differences because of the different amounts of pan cells that are found in there. So if you think that the pan cells are secreting important paracrine factors, changing those with age regionally might also affect the clonogenicity. It's something on our to-do list to kind of go back and see how they're different in the matrigel and the culture, but we haven't gone back to that. But I think that's a very important question, yeah.
Okay, thank you.
Mm-hmm.
You talked about how, like, after the fasting, you would kind of see like accelerated cell growth.
Yeah.
And I was wondering how did that cell growth compare, like, timeline-wise? You know what I'm saying? Like, did they catch up to and surpass the non-fasted cells, or did they just kind of catch up and be at the same rate?
Yeah, that's a very good question. So to address this, we're doing some cyclical fasting experiments and kind of like some timed experiments in the fasting, and then labeling with beer view and seeing how much the proliferation is affected under like the timeline of different fasting and then a timeline of different eating. So hopefully we'll have that information soon. But Yeah, they should eventually catch up. And again, you can only pass the mouse for so long before it's kind of on the other end of the bell curve and you're starting them, they're, really poor shape. So, yeah, but I think we're doing kind of cyclical fasting to address whether that affects any of the proliferation any different than if you have like an ad libitum mouse or like a one-time fasted mouse or so forth. Is there like a cumulative effect? And, so I think it's a lot about the timing as well. But in the caloric restriction, it's really interesting, right? Because you have actually a decrease of villi, and that makes sense, right? They're adapting to the fact that they don't need to spend so much energy on absorbed cells, and they're sort of priming themselves and increasing their stem cell pool. And then when you have the kind of repeating, I think that's when the villi really elongate. And in a lot of high energy diets, people observe like a huge elongation on villi, particularly in the proximal region, which is very important for taking up nutrients and absorbing a lot of fats and also glucose. And another interesting aside, in the aged animals, you actually also some people observe like elongated villi depending on the region, because again, maybe they're trying to overcompensate, and some of them have longer intestines as well. So maybe that's like a compensatory mechanism. So there's a lot of moving parts and a lot of regional differences as well to take into account. Yeah.
So I don't think you addressed this in your talk, but what is known about, so like what mouse strain did you use? And if I took a panel of mouse strains, different backgrounds, and fasted them and just measured, the how quickly they proliferate, what is like the range of variance just across mouse strains?
Yeah, so that's a good question. We mostly work with black 6 mice. I think other colleagues that have repeated some of these experiments have tried in various different strains like B129s, and I think they've seen similar things. But we haven't, I think that's a great experiment. Actually, when I was a postdoctoral fellow, I wanted to grab mice from other facilities and other strains to do that experiment, and I never got to do it, and I really should go back and do it because I think the variance will be very important. And it might correlate with differential expression of some of these transporters or other important factors, that kind of sense the nutrients and propagate, the important them. So yeah, it's a very important aspect because a lot of these strains are so inbred, so we should try some of the popular strains.
All right, thank you very much.
Thank you.